Alrighty. Let me turn the lights down. Get our screen on. All right. So on Friday, our main focus was our steroid-based hormone pathway. And so that's what you did the cut and paste activity for. Today and tomorrow, our pathways are going to be the protein-based hormone pathways. And their receptors, because proteins are going to be polar, their receptors are going to have to be in the cell membrane because polar things can't pass through the membrane unassisted. So today, we are doing the G protein linked receptor pathway. And so the main hormone that we'll be using for this pathway will be epinephrine. And epinephrine is responsible for bringing out your fight or flight response. So when you're in an emergency or you're scared or you're getting ready to fight somebody and your heart rate speeds up and you start breathing a little bit heavier, it's epinephrine that does all that. Epinephrine also is going to activate enzymes that can break down glycogen in your liver, which remember is our sugar storage form. And so epinephrine is going to trigger glycogen to be broken down in your liver to produce glucose, because if you're in an emergency and you're getting ready to fight something or run away from something, you're going to need that extra fuel, and the glucose is the fuel for your muscle cells. The pathway we're doing tomorrow is receptor tyrosine kinases. And so if the names sound a little bit intimidating, yeah, I know. But I am breaking it down just as easily as I can. And that's why we're drawing it step by step. Because if I just gave you notes on it and didn't draw the pictures with you, you wouldn't be able to follow this pathway step by step. Receptor tyrosine kinase pathway is a lot shorter. This is the pathway that insulin takes. The G protein pathway is one that a lot of medicines are based around. It's also the one that a lot of bacteria tend to affect, like Vibrio cholera, which causes cholera. It will affect the G protein pathway. Receptor tyrosine kinases, which you want to associate them with, are cell-specific anti-cancer medications, the ones where you don't have to, take a chemotherapy drug that causes you to lose your hair and make you feel yucky all the time. These are like genetically cell-specific anti-cancer medications, and they're going to affect that pathway that we do tomorrow. And then finally, we have ion channel receptors, and that's really just kind of like what we did with synaptic signaling. So we don't really do a whole lot more with that. Okay, so a little bit of background information on this next slide. Guys, where are you supposed to be in the walk? Okay. All right, peeps. So first of all, what's a G protein? G protein stands for guanine nucleotide binding protein. No, you don't have to memorize that, but I think it also helps to know that the G is for guanine, which is one of your nitrogenous bases. A guanine nucleotide would be like guanine and a sugar and a phosphate group. So it's a guanine nucleotide binding protein. And what does that mean? It means it's a protein that binds to guanine nucleotides like GTP and GDP. So that's kind of similar to ATP and ADP, except ATP was adenosine triphosphate, so this is going to be guanosine triphosphate or guanosine diphosphate. So very much like ATP or ADP, but using GTP instead. So why do we care? Because it functions like a molecular on-off switch. And so like I said, you probably don't realize just how many pathways out there affect, or how many medicines out there affect this pathway, how many bacteria, may affect this, or their toxins may affect this pathway. And you might say, well, I've never heard it before. How important could it be? Well, it's kind of complicated. So I really doubt you have heard of it before you get to AP Bio. But if you go into any sort of medical research or cancer research, pathways we do today and tomorrow would definitely be something you need to be very knowledgeable about. All right. Last bit of background info before we get into the pathway. Okay, so G proteins can exist in two different states. They have an inactive form and an active form. In their inactive state, they have GDP bound to them. So just think about it this way. We know that GTP would store more energy than GDP, because remember, in a molecule like ATP, we know that energy is stored in the bonds between the phosphate groups. And so this has already had one phosphate group broken off. So it's not going to store as much energy as GTP. So this is our little G protein. This is our G protein linked receptor. This is a different enzyme called adenylyl cyclase. If GDP is bound to our G protein, then our G protein is in its inactive state. If GTP instead is bound to our G protein, then it's going to be in our active state, and we're going to show that by kind of having this little yellow halo around it, and that's how you're going to know that that G protein is active. All right, let's get into our pathway. So first, a signaling molecule or hormone binds to a receptor protein. That specific signaling molecule or hormone in this pathway, in our example pathway, is going to be epinephrine. Of course, it doesn't have to be epinephrine, but it could be. So I'm going to use the same colors throughout. So I would suggest you doing that as well. This is our G protein linked receptor. And this is going to be our signaling molecule or our hormone. And then just so we know where this is happening in our cell, we're going to draw some phospholipids, because that's going to tell us that this is happening in the cell membrane. So I know I'm writing over G, but it's a G protein linked receptor. So maybe I'll do it this way. Do that, and this will say G protein linked receptor. And then I can draw my phospholipids. Okay, so again, this is today and tomorrow we're talking about protein-based hormone pathways. They're not quite as generic as the steroid hormone pathway is. And so the receptor for these protein-based hormones is going to be in the cell membrane because protein-based hormones are polar, and polar things cannot get through the membrane unassisted. And we're not really trying to get that signaling molecule into the cell. We're trying to get it to have an effect on something going on in the cell, but I don't actually need epinephrine to get into the cell. I need its chemical message to get into the cell. So that's the first step. Second step, the binding of that signaling molecule to the G protein linked receptor is going to cause that G protein linked receptor to change shape to make room for the binding of the G protein. So notice all that happened is in this lower corner of that receptor protein, there's now a notch cut out of it, and that is where our G protein will bind. So you know how I'm going to tell you to study this, and I would not wait till tomorrow night to study it because you're learning a different pathway tomorrow. I need you to go home and master this tonight, and how you master it is by going through this and drawing it out step by step and talking your way through it, and then checking it against your notes to see what you missed. But I wouldn't wait till tomorrow night. We're doing a different pathway tomorrow. Okay, so first thing that's a little confusing. This big blue structure is called a G protein linked receptor. It is a receptor protein for the binding of this signaling molecule, which in this example is epinephrine. Okay, but it is a G protein linked receptor because where we carved out this notch, that is where a protein called a G protein is going to bind. So it's a G protein linked receptor because a G protein can bind, and it's a receptor for a protein-based signaling molecule like epinephrine. All right. Okay, so a G protein with GDP bound to it binds to the G protein linked receptor in the cell membrane. So notice our signaling hormone is still bound to it here. This is going to be our G protein. And it is going, move that up there. It's going to have GDP bound to it. All right, so once that signaling molecule binds to this G protein linked receptor, that will allow this G protein linked receptor to change shape and make a little notch carved out of it for the G protein to bind. Now, the way I kind of think about this step is kind of like if you were borrowing somebody's Chromebook charger or phone charger, okay? Like, to me, that's kind of what you're doing to the G protein. Because when G protein has GDP bound to it, is it active or inactive? It's inactive, but it wants to be active because it wants to do its job. Well, the only way it can get activated is when it's bound to this G protein linked receptor. So when the signaling hormone binds, it causes this to change shape to carve out a little space for that G protein to bind. And the G protein binding is like you plugging your phone up to somebody else's charger because you need it to, have more charge on it, right? And so that's kind of going to bring about our next step. Now notice the phospholipids. This is still happening in the cell membrane. Okay, so next, the GTP replaces GDP attached to the G protein. Now the G protein is going to be activated. I know that sounds weird because logically you could be thinking, well, couldn't we just add a phosphate group to GDP and then we'll have GTP and then it'll be activated? Why does it replace it? I don't know. I didn't make up this pathway, but that's actually what happens. GTP comes along and kind of kicks GDP out and takes its place. And because that G protein now is going to have GTP attached, now it can be activated. And so the only way that the G protein can become activated is if it now is going to have GTP attached to it. And apparently that only happens when it is docked in with the G protein linked receptor. So we said GTP is going to come along and kick GDP out. And so we're going to say that this is GTP there now. And then the way we're going to show that the G protein is now activated is by drawing, like if you have a yellow highlighter, I would use that to kind of draw a ring around this. And that's going to show that your G protein is now active. I'm going to draw in a couple phospholipids just to remind you that this is all still happening in the cell membrane. So far, so good? All right, now think about the G protein. The G protein is really just using this blue G protein linked receptor as like a docking station. It's really just using it so that it can come in and when it's bound here, then GTP can replace GDP and this G protein becomes activated. So think about, your electric cars. If you take them on a long trip, you've got to go to a charging station. So my sister lives in Florida. She just got one, some fancy electric car, and she had to stop like 3 times on the way from Florida to here to charge her car up. And so you can kind of think of this G protein linked receptor as like that electric car charging station, and you have to dock your car in there so that it can get charged up. Good? Let's keep going. All right, well now the G protein is activated, so it's done using the G protein linked receptor. Just like my sister's car, once it's fully charged, she doesn't need to stay at the charging station, so she can leave. Okay? So, our signaling molecule is still there, but our G protein has said, see ya, and it is detaching from the G protein linked receptor. GTP is still attached, which is why this is still activated. So we want to draw our little yellow halo around there. And remember, this is all still happening in the cell membrane. All right, now what you're going to see about this pathway is going to be that we have to, before we can see what the G protein does next, we've got to take care of this. Because knowing what you know now, the steps we've gone through, what really is the purpose of the signaling molecule binding to the G protein linked receptor? To make a space for the G protein to bind so that it can become what? Activated, right? And it's activated form. Exactly. So now that the G protein is activated and it's detached from that G protein linked receptor, do we still need that signaling hormone? So think about in this pathway, I know you want to go straight to what the G protein does next. I want to see what its next adventure is, right? But we can't, because it's kind of like when you're home and your parents fuss at you about leaving a light on in a room you are no longer in. Okay? They're going to go tell you to shut that light off. You're not in the kitchen anymore? Go shut the light off. So we're kind of shutting the light off here. We have to turn things off before we can proceed. And that's going to be kind of a common theme in this pathway. So the signaling hormone is going to detach from the receptor protein. Now the notch is still technically there. It will go away in the next step. And that's all I'm going to draw for that one. All right. So what is going to happen to this G protein linked receptor once the hormone unbinds? It's going to go back to its original shape. So that's the next one. And again, not going to draw a whole lot for this one. We don't need to. We're just showing that the G protein linked receptor reverts back to its original shape. Okay, so these last couple steps were like shutting the light off when you leave a room. Okay, now that the G protein is activated and detached from the G protein linked receptor, we got to take care of that before we can see what that activated G protein does next. So after the activated G protein detaches from this G protein linked receptor, the hormone or signaling molecule is going to unbind, and that's going to cause that G protein linked receptor to go back to its original shape. Now we can pick up with what our activated G protein is up to. Still in the membrane, by the way. We have not entered the cytoplasm yet. Okay, so next the G protein with its GTP attached binds to an enzyme that is also embedded in the cell membrane called adenylyl cyclase. All right, so let's show that. All right, this is going to be our enzyme called adenylyl cyclase. And it is also embedded in the cell membrane. All right, so our activated G protein with its GTP attached. We know that when it has GTP attached to it, we know that the G protein therefore is active because it's got GTP attached. But because that G protein is activated, when it binds to the adenylyl cyclase, that also activates that enzyme and turns it on. So we've got to show that adenylyl cyclase, I want you to do a yellow halo around it, because once this activated G protein binds to adenylyl cyclase, now adenylyl cyclase is active as well. So you need to show a yellow halo around both the activated G protein and the adenylyl cyclase enzyme that it is attached to. Then I'll draw a few phospholipids over here, just reminding you that as of right now, we are still in the cell membrane, but that is about to change. So if you don't know this, you probably have heard this in bio, but when something ends in ASE, that's a pretty good hint that it's an enzyme. Okay, so adenylyl cyclase is an enzyme. And enzymes catalyze metabolic reactions, or they speed up chemical reactions. They help things to occur faster than they would on their own. All right, let's move on. And we will draw this one, but we got to fill in our notes for it first. All right, so it says that adenylyl cyclase enzyme converts ATP to cyclic AMP. Cyclic AMP stands for cyclic adenosine monophosphate. And so we know what ATP is. We know it has 3 phosphate groups, an adenine nitrogenous base, and a ribose sugar. Cyclic AMP basically chops, it's going to have 2 of those phosphate groups removed, and it makes this bottom part into like a ring. So that's why it's called cyclic AMP. We've mentioned cyclic AMP on Friday when we talked about the generics of a protein-based hormone pathway, and we called cyclic AMP a very important second messenger. Okay, second messengers are needed when you are transitioning from a series of steps occurring in the cell membrane to things happening in the cytoplasm. So this ATP converting to cyclic AMP is happening in the cytoplasm or on the cytoplasm side of the membrane, and it's going to be adenylyl cyclase that is catalyzing that reaction. And so that's why we need cyclic AMP, because it's going to be our first messenger inside the cell. It is our second messenger overall. Our first messenger was epinephrine. So you have to know, this is like a word association. When you see cyclic AMP, you say second messenger. There are some things in AP Bio that just, you got to be able to associate those words together, and that's your first one. Cyclic AMP is a second messenger because it's going to be involved in one of the first steps that occurs in the cytoplasm after a series of steps happening in the cell membrane. So cyclic AMP is small, it's not a protein, and it's water soluble. You just really need to know it's a second messenger. So we're going to draw this step on the next page. There we go. All right, that adenylyl cyclase enzyme is activated. And the reason that it's activated is because it has the active G protein attached to it. So I'm sorry if I'm in your way. I'll move so you can get this down, but you kind of know what this stuff looks like. This is not new. And because GTP is attached to that G protein, we know that that G protein is active. So far, this looks like the picture on the last slide, but it's different. All right, so know what we're looking at on each side. This is the cytoplasm down here. And this is the extracellular fluid, the fluid outside the cell up here. All right, so adenylyl cyclase is going to be converting. So this is kind of how we show one thing being converted to another. And I make the middle part attached to the enzyme that is catalyzing that reaction. So adenylyl cyclase is converting ATP in the cytoplasm to cyclic AMP in the cytoplasm. All right. So we definitely want some cyclic AMP made here, but this is a metabolic pathway, so we don't want this pathway to stay on forever, okay? Just like before, we compared to having that signaling molecule or protein-based hormone unbind from the G protein-linked receptor. We were basically saying, okay, we're done with that part. I need to turn that part off, and how I'm going to turn it off is by having the protein-based hormone or epinephrine unbind from the G protein-linked receptor, and the G protein-linked receptor can go back to its original shape. Here, we are making cyclic AMP. Cyclic AMP is going to promote the rest of the steps happening, okay, which is going to bring us to our endpoint, which is basically us making an enzyme that breaks down glycogen in our liver to produce a lot of glucose that can be the fuel for our fight or flight emergency. But we don't want too much cyclic AMP to be made. So we got to shut this part of the pathway down. Once we've made our cyclic AMP, we've got to shut this off before we can continue on and see what wonderful adventures cyclic AMP will be on next. So make a mental note to yourself. We gotta shut off the switch when we leave the room. So make a mental note at the 2 points in this pathway where you have to go back and shut things off before you can proceed. Okay, so the G protein is going to act as a GTP-ACE enzyme to catalyze the breakdown of GTP into GDP. Again, I know that sounds weird because I know you say, couldn't we just take a phosphate group off of GTP and have it become GDP. I know, that would make a lot of sense. That's not what happens. Okay? The G protein acts as a GTPase enzyme and breaks down, it itself helps to break down GTP into GDP. Why is that significant? What's going to happen when I now will have GDP attached to my G protein instead of GTP? It's going to inactivate the G protein, okay? And the G protein right now is attached to what? That adenylyl cyclase. And that's why we said it was active, because the G protein was active. So when it bound to the adenylyl cyclase, it activated that. Well, guess what? If this becomes inactive, adenylyl cyclase is going to become inactive. And that means no more converting ATP to cyclic AMP. So let's try and show this as best we can. All right, so it is going to act as a GTPase enzyme, and it is going to convert GTP into GDP. And that will inactivate the G protein. And right now, the G protein is still inactive, or the G protein is inactive. That will inactivate the adenylyl cyclase enzyme. Okay, just reminding you that this stuff is all still happening in the cell membrane. Okay, so the G protein is now inactive again. So we can even write, if you want to write that G protein acts as its own GTPase enzyme, you can. And that's why it's converting GTP to GDP. Okay, so the G protein is now inactive again, and it's going to detach from adenylyl cyclase. Going to have its GDP attached and no yellow halo. And essentially, that adenylyl cyclase was kind of like using the G protein. Because when the G protein was active, then adenylyl cyclase was active because they were bonded together. And so now, the G protein is no longer active, so it leaves and detaches from adenylyl cyclase. So kind of a long shot example or analogy, but it's kind of like if you had a celebrity couple that were together and became famous. So this one was famous, and by dating this one, if that one became famous, and then this one fell from grace or got canceled. Okay, so they were no longer in the public eye, and so they're going to detach, and therefore this one's not going to be in the public eye either. That's kind of a crappy example or analogy, but you do what you have to remember the steps of this pathway. Okay, so G protein has detached from adenylyl cyclase. So adenylyl cyclase is no longer active. That's a C, by the way. That is adenylyl cyclase. And it is no longer active. What was adenylyl cyclase's job anyway? It converted what to what? ATP to cyclic AMP, but that's only when it's activated. So now that adenylyl cyclase is no longer activated because the G protein is no longer activated and no longer bound to it, now no more cyclic AMP is going to be made. We've shut that pathway down. Okay? So again, we've had 2 points in this pathway where we've had to stop and shut the pathway off, shut down this step before we can proceed. And so just be familiar with what those 2 steps are. All right. Next diagram. All right. This one's important, big picture wise. We can memorize the steps all we want, but you got to understand the big picture with this pathway. And this is trying to show you amplification. So the numbers off to the side here are how many of each thing there typically is. Okay, so you've got one epinephrine binding to one G protein linked receptor. Everybody with me so far? All right. Then we've got one G protein, then we have an activated G protein, and there's our adenylyl cyclase. You've still just got like one of each of those. And what reaction did you tell me adenylyl cyclase catalyzes? What to what? ATP to cyclic AMP. So we see that happening here. Now notice when we read over from cyclic AMP, that's 20. Okay, what that means, remember from your biology class that enzymes are reusable. And so that one adenylyl cyclase enzyme, once it's activated, can convert multiple molecules of ATP to multiple molecules of cyclic AMP. So just by having one of each of these, the fact that that enzyme is reusable, we make, let's say, about 20 cyclic AMPs for every one epinephrine we start with. Okay? And then cyclic AMP, when you see something like this, we call this a phosphorylation cascade. Okay, which means we're kind of like adding or taking away phosphate groups to activate the next thing in the pathway. So once we make cyclic AMP, notice from the picture, remember we always want to look at the pictures, they're there to help you. Cyclic AMP converts inactive protein kinase A to active protein kinase A. Now again, not going to make you write all these steps out on your quiz or on your test. I have no interest in that, but I am going to have you put them in order. So you've got to remember that we've got to shut down the enzyme making cyclic AMP before we can pick back up with cyclic AMP and see that it's going to convert inactive protein kinase A to protein kinase A. Then protein kinase A is going to activate this enzyme, which activates that enzyme, which this is your endpoint. Okay? This is what we want. The final enzyme in the pathway is going to be glycogen phosphorylase, and that enzyme converts a bunch of glycogen to glucose. Remember why we need that. We're in our fight or flight response. We either got to fight somebody or we got to run away from somebody. Okay? Our muscle cells are going to need a lot of glucose to do that. Notice how many. You're making like 10,000 molecules of glucose for every one epinephrine you start with. Okay, so we say that this pathway gets amplified as it goes because, yes, you have one of everything to start with, and then each adenylyl cyclase maybe makes 20 cyclic AMPs, and then each cyclic AMP makes so many protein kinases, and then each protein kinase can activate like 5 phosphorylase kinases. So the message gets bigger as you go down. That's what it means by amplify. All right, let's talk a little bit about our enzymes. Okay, so protein kinases transfer phosphates from ATP to protein in a process called phosphorylation. All right, so we know ATP, we'll say, is rich in phosphate groups because it has 3 phosphate groups. And so you remember that character Robin Hood that would like steal from the rich and give to the poor? To me, kinases are a lot like Robin Hood. They are going to steal phosphates from a molecule that has several phosphates, and they're going to typically put them on a protein that doesn't have a lot of phosphates. Okay, that's what kinases do. They are like the Robin Hood of the molecular world. Okay, and when they add a phosphate group, or when they take a phosphate group off of ATP and add it to something else, adding that phosphate group to something else is called phosphorylation. Now, the opposite of that is going to be protein phosphatases. If you're removing a phosphate group from an organic molecule like a protein, that is called dephosphorylation. And that is basically the opposite of phosphorylation. All right. And then the thing at the bottom of your page, you're just going to scratch out. So just scratch through that thing at the bottom of the page. Okay. So last we left our pathway, we were shutting down adenylyl cyclase because it had already made as much cyclic AMP as it was supposed to. And so we had to shut it down so it didn't make too much. So now we're going to pick up with cyclic AMP that was made by adenylyl cyclase and see what it's going to do next. And I kind of gave you a sneak preview of it on that diagram that we looked at. Okay, so cyclic AMP activates protein kinase A. This is kind of like a chain reaction. Protein kinase A is going to activate phosphorylase kinase. Again, you don't have to be able to write out these terms because you're just going to be putting them in order. All right, so let's flip to our next page. We are almost done. All right, phosphorylase kinase activates glycogen phosphorylase, which breaks down glycogen into glucose. So remember, we said this is our endpoint. Our endpoint means we should make about 10,000 molecules of glucose. And that's going to serve as fuel for our muscle cells so that we can fight off whoever we need to fight off, or we can run away from whatever we need to run away from. Now we have our cellular response. So our cellular response, the end of this pathway, is glycogen getting broken down into glucose, and that's if epinephrine, adrenaline is the original hormone. I don't call it adrenaline a whole lot in here because epinephrine is the hormone name. Now, what we like to do a lot with this is do a lot of what ifs. Like, what if you had an enzyme that blocked the production of cyclic AMP? Well, if you don't make cyclic AMP, you're not making a bunch of glucose because the pathway would shut down right there. Okay, so this is a long pathway because every step has got to happen for you to make these 10,000 molecules of glucose that can serve as fuel to your muscle cells. So if you have something that inhibits a step, you have to think about how this endpoint would be effective. All right, so we talked about this being the amplification of the original signal. This is also showing you that one adenylyl cyclase can activate multiple cyclic AMPs. Each cyclic AMP typically only activates one protein kinase, so that's why I think this was 20 and that was 20. But then each protein kinase can activate multiple phosphorylase kinases, and each phosphorylase kinase can activate multiple glycogen phosphorylases, which we know break down glycogen to glucose. Oh no, I'm not doing that. Well, maybe I am. Okay, so G protein pathways are important in embryonic development, vision, and smell. This is where they're really important. All of those disorders that we've listed there are caused by bacteria. So cholera, which you'll be investigating tonight, whooping cough, botulism, which is a form of food poisoning, but you read about that with Botox. All of these bacterial toxins that they make interfere with this G protein pathway, and that is how they bring about their effects on your body. So 60% of all medicines used today exert their effects by influencing G protein pathways. I might add in something right below that, so don't put those notes away yet. Hold on. Yeah, we're going to add this in below it. So you're just writing this in right below that last slide of notes. And so remember, I told you a lot of interpreting this stuff is going to be things that inhibit or reverse certain steps. So there's an enzyme called phosphodiesterase, which converts cyclic AMP to AMP. So it gets rid of cyclic AMP. And if you don't have cyclic AMP, you're not making 10,000 molecules of glucose at the end. And so some drugs are phosphodiesterase inhibitors, like nitric oxide. Hold on one second. I just want to look at this picture. All right. All right, that's where we're going to stop. We're not going to do those last two written things down there. I'm going to show you a video animation of this pathway, and then I'll give out your cholera case studies for you to start on. We've got about 10 minutes left. Sorry, I'm just trying to figure out which one I used to have. Is it this one? So basically what you're seeing is the cyclic AMP that was made from ATP activates protein kinase A. And then when cyclic AMP binds to protein kinase A, you're going to make that next enzyme in the pathway. Well, you're going to make this, which is going to interact with the next enzyme in the pathway, which is phosphorylase kinase. That's the last enzyme in the pathway. Okay. So I know that that animation may have confused you further, but I'm trying to tell y'all when you learn something complicated,